So today we are starting at this set of presentations and discussions on water quality and human health. So how does water quality relate to human health? There are beneficial effects from water and the oceans related to the direct effects from the environment and related to the high quality food that we get from water. There are also negative effects, which include harmful algal blooms, infectious diseases, deoxygenation of the waters and hypoxia, and so on. So if I begin with the positive effects, let's think about something as simple as going to the beach. Now, you may think that you do this for the fun of it if you take a trip with your family or friends to the beach. But in a recent article for the common people on environment and health related topics, a journalist, Jessica Booth, listed some 10 health benefits that accrue from going to the beach. And they are listed here. That includes vitamin D from the sunshine, reduced pain, better sleep, better lungs, reduced depression, and so on. You may argue that some of these benefits arise from just being outdoors and being active, but some of them are indeed related to the specific properties of seawater, such as the anti-inflammatory properties. They call it salt therapy related to salt in the seawater, or to the smell of the oceans, which are uh, rich in negative ions, the atmosphere above the seawater. So whatever reason you might go to the beach, you do want to make sure that water quality will be one of your criteria for selection for where you might want to go. So in this very simple context, and if you recall previous lectures, especially the one from SON, you might have learned, or you should have learned by now, that there are several indicators of water quality that are available to us through remote sensing from satellites. And of course, chlorophyll concentration is considered a good indicator of water quality in many ways. And we have heard a lot about water quality. We've heard a lot about chlorophyll from satellites. But there are other properties such as the diffuse attenuation coefficient, turbidity, water color, water clarity, suspended sediment load, the euphotic zone, and so on. And just about all of them are available to remote sensing. And the advantage of using a satellite-based method is that you can look at this problem at multiple scales. You could do it at the global scale. You can look at it at the regional scale, or you can look at it in a very local scale, depending on the problem that you are dealing with. You have a choice. But let me recall something that we have discussed in earlier lectures, and that is when you come to regional and local scales, and the more you shift to using visible spectral radiometers that were designed for land applications, you begin to make compromises and the solutions become more difficult. And it is always useful under these circumstances to have local observations for algorithm development or for validation. And it's also important to test various steps, atmospheric correction, in-water algorithms, and so on. And I think you will find that you never have enough data. So if somebody can get more data to improve your methods, the better it would be. Now, Gemma next week will discuss these things in more detail. But the other health benefit from the sea are related to food. We know that we can derive high quality, high protein food from the sea, mainly as fish and seafood. But the fish or seafood are also a source of certain essential um, fatty acids that are not produced by the fish themselves, but they are derived from phytoplankton. Now, I don't know about you, but when I was little, my mother used to force me to drink shark liver oil. Terrible taste, terrible smell, but I had to drink it, my brother too, because it was supposed to be good for you. The reason lies in these essential fatty acids. But now there are actually products that you can buy that are sold as being helpful for longevity. They are superfoods. And what are they? You're bypassing the fish and you're going straight to the marine phytoplankton, which produce the fatty acids, which are then um, consumed by fish, and then you get the fatty oil in the fish. So according to Amazon, you can buy 100 grams of this superfood derived from phytoplankton if you have 100 pounds and more of cash. So if you then go back to the fish, fisheries production and the revenue from fish depends on primary production by phytoplankton, which depend on and of course the fish yield will depend on the magnitude of primary production and the phenology, which is the time sequence of the seasonal events in the phytoplankton dynamics. The primary production also depends on water quality. So we are back to water quality, and the water quality indicators or aspects that are relevant for primary production include light penetration, water quality, water clarity, and water color. I'm not going to go into details on this, but you will hear more details about it further down this training course. Bob Bruin will be talking about primary production, and Dionysius Reitsos will talk about indicators of ecosystem status. Now, I did mention the essential fatty acids. So these essential fatty acids are considered to be essential, and that's why they're called essential fatty acids, for health and survival of vertebrates. Now, different kinds of classes of phytoplankton produce different types of these fatty acids. Some are produced exclusively by diatoms. Some of them are produced, diatoms also produce another one called the EPA, which you may know more familiarly as an omega-3 fatty acid. Eggs are supposed to be rich in those omega-3 acids. There are concerns now that as fish communities change their distribution and abundance, there could also be a link to the availability of these essential acids from the ocean. Now, you may be surprised to know that there are methods to monitor some aspects of the distribution of these essential fatty acids from remote sensing. In the work that I am referring to, we went from chlorophyll and diatoms to a relationship between diatom and the fatty acid EPA to map the EPA in these regions. The interest was to see if the EPA production by phytoplankton is sufficient to meet the increasing demands for EPA from the oceans. Now, supporting fish yield from the ocean can also be achieved through support to fisheries operations, where one can use satellite-based methods to predict areas where you might find rich fishing. The object, and many countries are doing that, and the objective, of course, is not to overfish or to help overfish, but to improve the efficiency of the fishing effort, catch your daily quota in less time and using less fuel. And then you contribute to sustainable, better management of sustainable fisheries. There are also ways in which you can use space technologies to support aquaculture industry. In fact, as we are fishing down the food chain and the harvest fisheries are struggling more and more, it's becoming an increasing problem to provide high quality food for the growing populations, and governments are turning more and more to aquaculture. So in support of aquaculture, you can use remote sensing in many ways, but some of them are listed here. They could be to detect harmful algal bloom and to provide warning to reduce loss from these events. It might be to predict hypoxic events. And in case you don't know, these are events where the oxygen levels in the water go down to such an extent that the animals find it difficult to survive and you can have mass mortality. You can use remote sensing to study the carrying capacity of a habitat. In this example here on the bottom right, you can see how an airborne remote sensing tool was used to look at the grafts of aquaculture cultured mussels and how you see the depletion of the mussels, of chlorophyll in the areas where the rafts are placed. And you can also use remote sensing back to water quality for site selection and for monitoring impact of aquaculture. Now, if we come closer to freshwater, the effects or the need for water is even more obvious to see. If you look through history, you will see that typically civilizations and cities have sprung up around sources of water. And a classic example of how water dictates our entire life is this city called the Fatehpur Sikri that was built by the Mughal Emperor Akbar in 1571. When they designed it, they paid a lot of attention to water. They had artificial lakes and they had water channels and they had drainage channels. But when the entire population moved there, eventually it became impossible to sustain the city because the water supply became insufficient from the spring on which they were dependent. So within 10 days, this entire city was abandoned. As somebody remarked in one of the papers I was reading, okay, in those days, if a city failed, you had enough land and maybe you had the money to relocate to another place. But today you have to be careful because land is in short supply, water is in short supply, and you have to make sure that we use water efficiently and intelligently. But water is essential for our life, for our survival, but water quality is also related to health. And this idea that water and air quality is related to human health is not a new one. If you take the example of the golden age of Islam, I think it went from the 8th century to the 14th century. They knew that the success of their hospitals which were called Bimaristans, depended on air and water quality. So they built their hospitals on hills or by rivers, and then their engineering was such that they diverted the water to flow through the courtyard and the halls of the hospital, and then the water flowed back into the river. Now, in the top right-hand corner here, I have the architectural drawing of one of the hospitals where, in fact, you see that at the very center of all the wards was a water tank with a water fountain. A famous physician, Abu Bakr al-Razi, was known to have carried out an experiment to determine air quality in Baghdad before he chose the site for a Bimaristan there. And the experiment he carried out was to place pieces of raw meat in different parts of the city. And he and his students monitored how fast the meat became decayed. And the assumption was that the place where the meat stayed fine for longest period had good air quality, and therefore was a good location for placing the hospital. In Islam, flowing water was considered to be good and incorporated into their architecture, whether it was for gardens, private residences, or the Turkish baths. It's interesting that if you look at the Indian Ayurvedic principles, they did not believe in flowing water, but they strongly advised boiling water before you consumed it. It's another good principle that we might adopt today. And now my slide is not moving. Okay, so I could summarize this part of the talk by saying that our life depends on water, not just our health, but our well-being in the broadest sense of the word, whether it is for recreation or for transport and for many riches that you might extract from water. In fact, it also affected our religions. Some of the religions in the historical times went from one country to another through transport, through ships. But water has many faces, and sometimes it can turn on its anger on us. And the example I have here is that of the 2018 floods in Kerala, in a region called the Vembanad Lake and surrounding areas. The impact was the worst. I understand that millions of people were displaced and over 400 people died from this one instance of a terrible flooding incident. But perhaps, no, sorry, at that time, the people did make use of flood mapping from satellites to understand the magnitude of the problem. This is a newspaper article of the time that showed satellite images from Sentinel-1 produced from Europe based on the ESA sensors. And they were a valuable source of information. But if satellite maps are to be used for effective management of response to floods, the risk maps should be timely, they should be frequent, they should be reliable, and they should reach the decision makers. And I don't think we are quite there yet in making best use of satellite information for reducing the damages from floods. And this problem will be dealt with in more detail again by Gemma next week. Now, I talked about this particularly disastrous flood, but I wonder sometimes whether the bigger problem is not the insidious, frequent, repeated occurrences of natural disasters. Again, I revert to a newspaper article from this region, and they say that this repeated flooding, torrential downpours, intrusion of saline water, and disasters such as these are wearing down the resilience of a part of our study area called Kuttanad, which is typically considered in commonly referred to as the rice bowl of Kerala. The floods caused tremendous devastation to the to the harvest of rice in that particular year. Though I also understand that the benefits of nutrients from flooding led to a bumper crop the next year. Anyway, the point I want to make here is that the incidence of repeated occurrences of natural disasters has led to climate refugees. Some 15% of the people have already left this region, which I find is a sad thing, but I also like to uh, point out that this is not a unique story. But what I want to say is that this leads me to say that the problem is urgent, and we need to address the resilience of the human communities to perturbations under extreme weather events associated with changing climate. So if you now look at the global picture, you see that what is happening in Kuttanad and Kerala and regions like that, it's not an region specific, it is not isolated. The water related disasters are now a global phenomenon. It's affecting people all over the world. And according to this review paper, the number of people affected run into hundreds of millions. And the health impacts of these water-related disasters are also important, and they continue sometimes long after the physical damages related to flooding have receded. So if you partition the types of diseases that follow a water-related episode, you see that waterborne diseases occupy a big part of the pie, and it is more important during flooding, but it is still important when you have droughts. Mental problems is a big component of the health problems that follow. So for much of what follows, we are going to look more at waterborne diseases. So what is the global relevance of waterborne diseases? According to WHO, some 200 million cases of malaria occurred in 2020, and associated deaths were over 600,000. If you look at the case of cholera, there were millions of cases reported, and somewhere between 20,000 or 140,000 deaths were associated with cholera worldwide. See this big uncertainty in numbers, and this is a problem. There are frequently high underreporting of waterborne diseases, so remedial measures become difficult. What if we change and look at the statistics for drinking water, because many of the diseases that we are going to look at are associated with contaminated drinking water. Again, according to WHO, some close to 800 million people lack even a basic service, and about 150 million people are dependent on surface water, are part of this group of people who don't have enough sustained supply of drinking water. And they say that even if you have drinking water, it could be contaminated. Some 2 billion people have no guaranteed contamination-free water supply. So contaminated drinking water can cause up to 500,000 diarrheal deaths each year. And according to WHO's prediction, in about 2 years, half the world's population will be living in water-stressed areas. And furthermore, as we know, provision of safe drinking water breaks down in extreme weather, such as flooding, and that is always frequently followed by outbreaks in diseases. But in case you think that I am talking about a third world problem or a developing country problem, think again. This is a recent article that appeared in BBC, and it talks about the problems with sewage in waters around UK. So these statistics I found to be staggering. Water companies who are charged to look after the treatment and discharge of sewage frequently are allowed to discharge untreated sewage into the sea and into natural water bodies. And so the culprits are here. And they add to more than up to about 2 million hours of raw discharge being disposed into rivers and the seas. And this has only made the news because surfers have united against mistreatment of our waters. So a group called, what do they call themselves? Surfers Against Sewage, I think, something like that. And they have been protesting against these, and all of a sudden it is in the national news. So we cannot underestimate the role that citizens can play in correcting some of the current problems. Now, these are all health problems, and we are at a course on remote sensing and water. So it might be fair to ask, what's it got to do with us? The reason is that many of the factors that determine health and the outbreaks of infectious diseases or its treatment lie under the direct purview of the health sector. The other sectors involved include those who are responsible for sanitation, water supply, and water treatment, as we just saw, education, agriculture, trade, tourism, and so on. But importantly, it also includes environmental and climate change as factors that determine health. And that is the factor that we are talking about here. So what we are interested in is in looking at what space technology can contribute and how can it be integrated with other sectors. Because by ourselves, we are not in a position to solve this complex problem, but we can contribute. So let's look a little bit more at the role of the environment in deciding public health. So in our context, the importance is the role the important aspect is the role that environment can play through waterborne and vector-borne diseases. Now, Stefano mentioned it. There is evidence that diseases associated with animals that are then transmitted to humans and that are referred to as zoonotic diseases are on the increase. So these diseases that are transferred from animals to humans can be classified as those that are transmitted by direct contact with the animals, indirect contact, or that are transmitted through some vector or the food that we consume, or waterborne diseases. And if we look at the waterborne diseases or water-associated diseases, they are a diverse group, and they include those that are diarrheal in their nature, such as cholera. There are pathogens that cause skin diseases. Then there are those that are responsible for vector-borne diseases such as malaria or dengue fever, and further, yet others such as hepatitis. Now, water fosters many types of pathogens or responsible for infectious diseases, such as the Vibrio cholerae bacteria, that is known to have many reservoirs in the water, and that is responsible for cholera. Mosquitoes are responsible for many vector-borne diseases such as malaria, dengue, chikungunya, Zika, and they require water for completing a part of their life cycle. Macrophytes, or large floating vegetation, which sometimes may be invasive in certain regions, such as the water hyacinth in Vembanad Lake. And they may be very beautiful, but they can create stagnant pools, facilitating mosquito breeding. And water hyacinth is also known to be associated with Vibrio cholerae, or as some other large floating vegetation. So let's take a little bit of a closer look at the genus of Vibrio that include a number of pathogenic bacteria species. Vibrio cholerae that we just encountered include some 200 serotypes, but only 2 that are referred to as O1 and O139 are associated with major cholera outbreaks. Vibrio alginolyticus causes wound and ear infections if you are exposed to contaminated water. Cholera bacteria, on the other hand, can only cause a disease if you actually drink the infected water or food. Vibrio parahemolyticus is associated with food poisoning, and Vibrio vulnificus is a deadly one that infects wounds. If you walk through contaminated water with open wounds, and if you are vulnerable, you can septicemia can follow very quickly, and the mortality rate is very high. In Baker, Austin's, and colleagues' very good summary, which I have shown here, they have classified all their properties in a very nice diagram. And if you look at the genome, the important point to note here is that these organisms are capable of recombining and with other organisms, and they also can do horizontal gene transfer, and that leads to the evolution of the species, and it can also lead to buildup of antimicrobial resistance in the genes if these DNAs are in the water. So in the environment, you can find them, these pathogens, in association with fish. They may be free living or free floating, whatever you want to call it, in the water itself. They may be associated with crustaceans, and they may be associated with algae. The transmission, it can be oral or through wound exposure, as I indicated here. And you can come into direct contact with the bacteria, for example, by drinking the contaminated water. And it can also be through person-to-person transmission. So if you take a look at the overview of the role of the environment in causing infection, sanitation conditions appear as a key problem, key factor. Environmental conditions that can dictate growth or decay, and then which in turn would determine how much bacteria are present in the water. And then these may come back to infect us, for example, through fish or seafood. The circulation and residence time and flushing rate of water bodies can also determine the growth rate and again influence the pathway to infection. So if you look at some of these things in one at a time, let's look at the environmental conditions and how they may affect growth and decay. So let's take temperature and salinity. It's been known for a long time now that temperature and salinity have an effect on growth rates of Vibrio. This example is for Vibrio vulnificus, and you can see that they don't like decreasing salinity, and they have some preferred temperature beyond which they don't survive too well. So this has been taken to further extent in some work. For example, Pfeffer and colleagues, they studied the Vibrio and temperature in North Carolina's estuarine waters. And they showed that there was a close relationship between water temperature and Vibrio genus, all the species in that genus, or if you take the Vibrio vulnificus by itself. And this relationship with temperature was stronger than many other environmental variables that they measured, such as total bacterial count, phosphorus, ammonia, salinity, and so on. There is a study that looked at the North Atlantic as a whole, and they showed that Vibrio abundance in the water increased with sea surface temperature. And using a generalized additive model, they showed that sea surface temperature and the Atlantic multi-decadal oscillation explained some 30% of the variance. And they did the analysis for the different types of Vibrio, Vibrio cholerae, Vibrio parahemolyticus, and vulnificus. And you can see how the different types of Vibrio were affected. In another very interesting study, Baker Austin and colleagues showed that there is an increasing incidence of Vibrio-related hospitalizations, I think, in Northern Europe, and that there was a strong relationship between anomalies in sea surface temperature and incidence of Vibrio infections in Finland and Sweden. They use satellite data to do this, and they also point out that these occurrences of positive anomalies in temperature, or known as heat waves, they are becoming stronger, and the frequencies of these events are increasing in recent years. So based on these analyses, one expects that vibriosis associated with these pathogens are likely to spread northwards with higher water temperatures. So we also looked at what others have said about Vibrio cholerae in the natural environment as a function of temperature and salinity. So this is a compilation from Takemura and colleagues, and they reported that Vibrio had a preferred range between 10 and about 28 degrees for high numbers of Vibrio to be present. In a lab-based study, Materna and colleagues also reported that they had an optimal growth temperature of 28 degrees, but they could survive between 41 and 18 degrees centigrade. So this is a map of the seasonal variation in temperature in Vembanad Lake, our study area in Kerala that Stefano also referred to. In blue, we have in situ observations, and in red, satellite-based observations averaged for the whole lake. So this is the optimal temperature for Vibrio growth as reported by Materna. And we see that the temperatures in Vembanad Lake are now already reaching very high values, maybe heat waves of up to 36 degrees. So we wondered whether that might push us beyond the upper limit of suitability for Vibrio, but people say, but Vibrio can survive very well inside the human body at 38 degrees. So it's not, we can't sterilize the Vibrio by letting waters warm up too much. So moving on to the aquatic reservoirs of pathogens, and again using cholera as an example. This is complex because Vibrio gets everywhere. Vibrio cholerae is a naturally occurring component of brackish and freshwater systems, and they are known to interact with phytoplankton, zooplankton, and they can exist as free living organisms in the water. And if fish consume infected phytoplankton or zooplankton, then the fish can transport the bacteria over long distances. If you look at their life cycle, it is generally understood that contact or consuming contaminated water or food can cause an outbreak of the disease. But in the interval between successive outbreaks, they have to survive in an aquatic reservoir. And this may be through phytoplankton or zooplankton or insect egg masses, exoskeleton, detritus. So a lot of things can harbor these bacteria in between infections. And once it is inside a human host, you fall sick. And then if more people come in contact with the shedding of infected material, for example, through stool, then it spreads and you have an outbreak, and they say that during an outbreak, you can have hyperinfectious vibrios and further spreading through contaminated material and so on. So back to remote sensing and using this information for predicting risk. There are several similar studies. I show here one by Demagni and colleagues. And they said that for an outbreak in Calcutta in India, they could predict the outbreak of cholera cases using satellite-based chlorophyll concentration, sea surface temperature, and rainfall data. And they used both in situ and satellite data. And they were able to predict the occurrences of the cholera cases with a 1-month lag. So the results are statistically significant, and the 1-month lag lets you develop early warning systems. So we can, if you take bring you back to the Vembanad Lake in Kerala, chlorophyll concentration is in principle readily available from satellites. But let me point out or remind you that if you are dealing with areas of persistent cloud cover, we also have to rely on in situ observations to fill the gaps. And we also need in situ observations to obtain information at depth. And I advertise Gemma's talk next week. So given all this, what is our understanding of the ecological link between phytoplankton and Vibrio cholerae? One hypothesis is that the link with phytoplankton or chlorophyll concentration from satellites is not a direct one, it's through zooplankton. So in this hypothesis, you will have a phytoplankton bloom, and then the phytoplankton bloom will attract zooplankton, the animals that are microscopic animals that feed on phytoplankton. And the zooplankton growth may follow a phytoplankton bloom by 1 to 2 months. And then Vibrio cholerae attach themselves to zooplankton in high densities. This is an electron micrograph showing Vibrio bacteria attached to, I think these are the mouth parts of a copepod, a type of zooplankton. And this is an electron micrograph showing a copepod carrying Vibrio. So the point is that by attaching themselves, they now begin to aggregate in high concentrations that are sufficient to cause outbreaks. And according to this hypothesis, there is a material called chitin contained in the carapace or the outer shell of zooplankton, and they serve as food for Vibrio cholerae. And there are many supporting evidences from the laboratory to show that chitin promotes the growth of Vibrio. But there are other studies that have shown stronger relationships between Vibrio and phytoplankton than with zooplankton. In this study by Asplund and colleagues, in their analysis of data from the southwest coast of India, they showed the strongest relationship between Vibrio cholerae actually and diatom abundance. Diatoms are a type of phytoplankton, a negative relationship with temperature, and also another strong relationship with chlorophyll. Copepod, the phytoplankton, the zooplankton that I mentioned, they had a less strong relationship. So they suggested that phytoplankton may provide resources that promote the growth of phytoplankton, the growth of Vibrio cholerae more so than top-down control by predators. They also summarize many reported mechanisms that can sustain the association between phytoplankton and Vibrio bacteria. In our own studies, Anas, who is in the panel today, and colleagues reported that in the Vembanad Lake, back to our Vembanad Lake, Vibrio cholerae, which is labeled here in green, was found more frequently in association with Vibrio cholerae than any of the other reservoirs that were examined, whether it was the sediment in red, macrobenthos, or animals in the bottom of the water, zooplankton, and water itself. So in this paper, they suggest that the Vibrio phytoplankton association could be related to activities of sorry, I'm getting tongue-tied, laminarinase and chitinase activities. Now, so this could support another hypothesis, which is that the ecological link between phytoplankton and Vibrio could be direct, and they could be through biofilms. Now, these are films that Vibrio cholerae can form around many substances, whether they are biotic or abiotic. And the formation of these biofilms are supposed to facilitate their access to nutrients and can also serve to avoid predators. But it need not be biofilms. It could also be, as we have seen, other ways of directly linking phytoplankton and Vibrio, but without necessary intermediacy of zooplankton. But here was another surprise for us. When we looked at the relationship between cholera bacteria, Vibrio cholerae bacteria, and chlorophyll concentration, we found that in some parts of the lake that we were studying, the relationship was positive. There are about a couple of hundred observations here summarized. And in the other part, the relationship was negative. So what could explain this observation? As I mentioned earlier, there are many results that show that phytoplankton can promote Vibrio cholerae. And the one explanation is that the mucilaginous sheets of phytoplankton can provide protection and nutrients to the bacteria. So these would be, along with biofilms and everything else, support the positive relationships that you observe. But there are studies that have also shown that some phytoplankton can have an inhibitory effect on the growth of Vibrio bacteria. In their experiment, Olafsson and colleagues showed that one type of phytoplankton called Prorocentrum micans promoted the growth of bacteria in a lab culture, whereas the addition of Skeletonema costatum belonging to the diatom group inhibited it, and the Vibrio completely died out actually. So these are dinoflagellates, I think, and this is certainly a diatom. So this kind of experiments might provide some explanation for why sometimes in the Vembanad Lake we might find a positive relationship and sometimes a negative relationships. I think I'm running desperately out of time, so let me pass quickly through this one to show that I mentioned earlier that satellites, I mentioned that floating algal vegetation could be an important factor in the story. And there are ways in which you can detect floating algal distributions using satellites. And some of them, at least one of them was proposed by Xiumin Hu and colleagues, and we are now beginning to apply these algorithms with modifications as necessary to map the floating algal vegetation in Vembanad Lake. So let me move on to the yet another condition that promotes infections, and that is poor sanitary conditions. So in one of our studies in the Vembanad Lake, we looked at the fecal contamination in Vembanad Lake, and using this bacteria, Escherichia coli, as an indicator of fecal contamination. Now, in the study, fecal contamination was well above acceptable levels, very high levels at all times, but they peaked during the monsoon season. This is a paper by, again, Anas Abdulaziz and colleagues. So when we looked at the average E. coli in the water against the rainfall, we found a strong correlation, which suggests that the rainfall is a factor that determines the water contamination by fecal material. Then when we looked at the cumulative rainfall against the cell concentrations in the water, we found that the peak appeared in July, and these are data collected in the same year as the floods, and the floods only occurred in August. So this indicates that the drainage of fecal material into water starts, well before any funding, and you can actually reach peak contaminations before flooding. And the other argument could be that when the floods are severe, the heavy runoff could wash away some of the contamination into the water. So in our current thinking, we feel that heavy rains and monsoon sustained rains, when they persist, the groundwater levels increase, and at some point they begin to reach the same level as septic treatment plants, allowing for cross-contamination of bacteria with groundwater as well as the rivers. So problem could be a serious one. Now, in many of these instances that I've been talking about, especially if we are talking about uh, current levels of bacteria detected through satellites, the satellites by themselves don't give us a predictive capability, for which we have to revert to mathematical models. And there are models that are available that are sometimes referred to as the SIR models, referring to susceptible, infected, and recovered components of the community. We have tried our hand at these models, and we found that we could do something half decent with COVID in Kerala, because compared with cholera, the COVID disease transmission is much more straightforward, with the predominant transmission route being from person to person. But we are still struggling with finding an effective predictive model for the role for waterborne disease such as Vibrio cholerae, something for some of you bright people to solve. Now, we've been talking about short-term predictions, and I want to spend my last few minutes by looking at the big picture and the predictions over years, shall we say, related to climate change. In this paper by Martinez, Urtaza, and colleagues, They studied the impact of El Nino on outbreaks of Vibrio-related diseases, and they were able to show a strong relation between El Nino events and outbreaks of diseases, Vibrio-related diseases in, I think this was in South America. Yes, South America. So they suggested that the occurrences of warm water across tropical Pacific associated with El Nino serves as a corridor for transport of Vibrio across long distances, causing outbreaks of diseases. So in this big picture, again, sea surface temperature appears as a key factor. In yet another interesting study, Escobar and colleagues looked at information on cholera bacteria from different locations, and then they analyzed the data along with satellite data on SST, PAR, or photosynthetically available radiation, salinity, and chlorophyll A. And in their map also, they come up with these long corridors of favorable conditions for bacteria to survive and do well. Then they took this brave step of looking at what would happen in the future as temperature predictions change. I mean, temperatures change according to climate projections. And their result was that depending on how we projected into the future, the areas that are susceptible to these infections can only increase in the future. The range of expansion of suitable environment may change depending on the prediction, but all predictions, according to their model, suggest a spreading of suitable conditions for these infections with increasing temperature into higher latitudes, northern and southern. And I want to now begin to conclude by mentioning the importance of data quality. Whatever analysis we might do will depend on the quality of the data. Now, satellite data are becoming increasingly readily available, freely and instantaneously, a timely manner. But it is extremely difficult to get some types of disease data for completing the analysis. Now, COVID-19 was a well-studied example of problems related to undersampling. And in this report in the BBC of a World Health Organization's report, they show the excess deaths associated with COVID-19 in different countries. And these are huge numbers. But they also say that all these countries, pretty much all countries, underreported the diseases. With respect to India, they said that 4.7 million people are thought to have died because of COVID-19, which is about 10 times higher than India's official records. And of course, India has disputed the claim by WHO. So the fact remains that if we can't rely on the diseases data, then all methods for environmental predictions of these diseases based on environmental conditions will be that much less performing. And of course, the data quality tends to be higher for infectious diseases such as cholera that we have been looking at. And data quality issues tend to be worse in low-income countries. And those are the countries where the burden of waterborne diseases are higher. And within country differences are also important. So we need highly spatially and temporally resolved data for some of these analyses to bear fruit. So these points that I have been talking about, they are related to the UN Sustainable Development Goals, and especially those that are related to water and those that are related to hunger, health, clean water, climate, and life below water. And there are many ways in which remote sensing can help. Now, SDG 6 is related to water and sanitation. And SDG 6 has the target of protecting, restoring water-related ecosystems, including wetlands, rivers, aquifers, and lakes by 2030. And we can't achieve such a target, which is just 7 years away, without understanding the role of bacteria and viruses that are responsible for infectious diseases. And as we have been talking now, and as you will see again in the future lectures, there are contributions that we can make through remote sensing. And there is a need for developing better geo-referenced risk maps. And you will hear more about geo-referenced data in Milton Campbell's talk in 3 weeks' time, I think. So let me just conclude by saying that remote sensing offers several avenues to explore the dynamics of pathogenic organisms, whether they are associated with waterborne or vector-borne diseases. But the problem is complex, and let me not leave you with the impression that remote sensing can do everything. It can't. So we have to work with microbiologists, geneticists, modelers, health workers, social workers, and so on to tackle the problem. So integration, communication, coordination, cooperation, they're all paramount. So as part of our effort to address this gap in integration, we are building a global network of interested scientists to learn from each other. And that one of those projects, a main project in fact, is called Onward, and which is also, along with Vigian that Stefano mentioned, is also a sponsor of this training course. So that is my last slide. And I quote a scientist called Thienemann. In 1925, he said, if you know the color of a lake, you also know its other characteristics. So once again, this is not anything new. Clever scientists knew that before. And let me stop there. Thank you very much for listening. Thank you.